Good morning. Right. So, as we decided last time, we will speak about the prokaryotes and eukaryotes. Right. I'm sure you have been studying this since long. Right. From 8th standard, you have been differentiating prokaryotes, eukaryotes in your examinations. Right. And then you have a list of features. Right. That are like prokaryotes this side. Right, eukaryote this side, nucleus present upside, this kind of. But we have to understand what does that mean, right? We have to understand what it is, right? So okay. So as the name suggests, prokaryotes are those which are pre, right? Pro is pre, karyote is or karyon is a nucleus, right? So those who are pre-nucleus, right, does not have nucleus. That means, right, are prokaryotes. Eukaryotes are those which have nucleus. So that's why they are called eukaryotes. So eukaryotes and prokaryotes, the primary difference which is given in their name itself is the nucleus is present in eukaryotes, the nucleus is absent in prokaryotes. Very clear, right? So that is given in the name itself. Now beyond, right? Now you know that okay, cell organelles present in eukaryotes, they are absent in prokaryotes. Right, size, one of the important part, right? The size of eukaryotes is bigger and the size of prokaryotes is smaller. So 1.5 to 100 microns, right, micrometer, that is the size of prokaryotes. Whereas eukaryotes range from 10 micrometer to 150 micrometers. Right, so obviously, of course, there are some eukaryotes which are smaller than the prokaryotes, but in general, prokaryotes are smaller than the eukaryotes. And this is the main differences that you definitely would know what is there, right? One thing is, of course, when we say there is no nucleus, that means there is a difference in their genetic structure. Right? What is the genetic structure of prokaryotes? Usually it's like a single DNA, right? So this is the bacteria. You have an epitope here, and then to which it is a DNA that is bound. That's a chromosome, bacterial chromosome or bacterial genome, right? That is a single DNA molecule. Most probably, right? Mostly this single DNA molecule is circular, right? Circular matlab kya hota hai? Two ends together, right? So they are concatenated. They are circular. They are concatenated. And most of the prokaryotes, this is the only genetic material that they have. So only one DNA, right? There could be some which exists like another circular DNA, small circular DNA, which are plasmids. What we call as the plasmids. How you define plasmid? They are extra chromosomal, right? Circular, self-replicating DNA. That is how we will define plasmids. Right? I'm sure you know that. Right? They are what? Extra chromosome. Extra chromosome का मतलब क्या होता है? They are apart from this chromosome. Apart from bacteria's own DNA, there are circular DNAs, self-replicating. Means what? They have a capacity to self-replicate. What does that self-replication means? Right? That means the chromosomes usually have one origin of replication site. What is that? One ori site. What we call it as origin of replication. So that is where DNA replication starts, right? So in eukaryote, on the other hand, there are bigger cells, right? They are bigger cells. Of course, they have a well-defined nucleus, right? And then there are cell organelles like mitochondria, right? They have, you could show mitochondria, right? Then you have ribosomes, right? You have ribosomes, right? And then they are cell organelles. There are vacuoles, right? Which are membrane-bound vacuoles, right? What's the difference between the biggest difference between prokaryote and eukaryote is like that, the membrane structure, right? What does that mean? Membrane structure. Membrane structures mean the entire cell is compartmentalized by different membranes. I always give a very common example. You know what, like when you are when you are fresh, right? Get degree and go out and get a job, right? Usually your salary is small, right? And then what would happen into that salary? How you make your living, right? You rent an apartment, most probably is like one room, right? And then that in one room you do everything, right? वहाँ पे भी bed है आपका, the same thing, you have a kitchen there, right? वहीं पे table है and then you study there, but then everything is into one small room. That is like a procure, right? It's a small thing, right? And everything in one compartment. See, there is no compartmentalization. But when you grow, your salary is increased, then what you can afford is like maybe 2 BHK apartment. 2 BHK is what? 2 bedroom, kitchen and hall, right? Means what? You have a well-defined kitchen, you have a well-defined hall, you have well-defined bedrooms, right? Will you ever go and sleep in kitchen? नहीं ना. और कभी किचन स्टोव बेडरूम में लेके जाके वहाँ पे कुक करते हैं? नहीं. Why you started laughing? Right? It sounds funny, isn't it? Right? Why it is funny? Right? Because see, the compartments is not just the room demarcations. अगर रूम में से आप बीच के walls हटा दो तो it's like one single unit, right? Right? But then what you are basically going to do there is a functional compartmentalization. See, it's not a physical compartment. Try to understand this difference, right? It is what? A functional compartmentalization, right? There is a defined space. Kitchen में kitchen का ही काम करोगे, है ना? So kitchen is where you cook, right? And bedroom is where you sleep, right? And hall is where you receive your guest, right? And then if you sweep those functions, you start laughing, right? When I said that, right? When you get a guest, you won't receive him directly in your bedroom, right? Unless it's like a very fast friend or something, right? So you won't like do that, right? So that is a functional demarcation. So what I'm trying to tell you that it is not just there are 3 to BHK differentiations between the apartment, but it's a functional differentiation, right? Kitchen has its own function, bedroom has its own function, hall has its own function, isn't it? Right? So in same way, eukaryote there is a functional demarcation. Right? In prokaryote it is not there. Right? In prokaryote it's like a single bedroom apartment or single one room accommodation. Right? Where everything happens in the same thing. There is no compartmentalization. Right? So DNA replication will also happen here. Protein synthesis will also happen here. Everything will happen into one single unit, one single space unit, right? That is how prokaryote is. And in eukaryote, what will happen? There are differences, and the differences are functional differences, right? That is what I am trying to stress on. Ribosomes will involve in protein synthesis. Mitochondria will involve into burning sugar and producing ATPs. Right? Endoplasmic reticulum has its own function. Right? So endoplasmic reticulum is also there, right? Which connects this in there. So endoplasmic reticulum, Golgi apparatus, right? So you have all membrane bound bodies there. Golgi apparatus, right? And Golgi apparatus are consistently releasing membrane vesicles, right? Those vesicles, when they acquire enzymes, they would become functional unit, right, of proteosomes, lysosomes, like that, right? So in eukaryote, and that's what you are like trying to do so far. Like now, I hope you understand the concept behind it, right? The nucleus present or absent, ribosome present or absent. Of course, ribosomes are there in prokaryote too. In fact, the ribosome, the sedimentation is low in prokaryotes, like in eukaryotes is high. So ribosomes are there, but otherwise also. So everything is happening there. Whereas there is only DNA there. And what is the DNA? What's the difference between this and this DNA? This DNA is complex with histone proteins, right? And makes itself into chromosomes. Right? We have seen that, right? How the DNA molecule end to end, right, bound with so many histones, compact, right? And then the compact thing is then become the chromosome. Why it has to be compact? Right? It has to be compacted because while replication there has to be a good separation. Right? So if you have a long DNA molecule and it get replicated, it would become double, right? And then to separate from one from each other would become a project in itself, right? Just to separate two chromosomes, right? Unless and until they are compacted, so they are contracted together, then you can. It's like you have wools, right? You know that for sweaters, if you want to knit a sweater, you take the wool, right? Right, and then the wool is compactly bound into a bundle. So bundles is fine. So you can segregate bundles, green bundle, yellow wool, right? And by different colors and texture, you can separate this wool, right? But then suppose there are no bundles and that wool is just kept into one carry bag. Now try to separate those. Now you will have a difficult time, right? You will spend an entire day detangling that wool because it's like so meters and meters long wool, right? So what you have to do is take some spindle and wound around it, right? Then it will become a bundle, and a bundle can be easily separated without getting entangled into it. Well, the same thing, right? What happened here? If this is DNA small, prokaryotic DNA is small, that's why it is easy to separate even in a binary fission where the cell is going to divide, right? What will happen to eukaryotes? Such a long DNA, then what will happen? If they get entangled into each other, we will never be able to separate those things, right? Never be able to separate that own replicated DNA, right? That's why it has to be bundled into chromosomes, and then chromosomes can very easily get separated, right? Without entangling into each other. Because I told you that DNA is ultimately as a molecule, right? Does not have a strength. Right, that okay, it's not an elastic that it would get stretched, right? It's not like that it will in itself would get compressed, right? It's not like, does not have physical property. So any shearing stress onto DNA will break it, and if the DNA breaks, you know that what will happen if the DNA breaks, right? That will be end story for that particular organism, right? If its DNA is broken. Right, so anything happen to DNA, even in prokaryotes also, in eukaryotes also, right? If the DNA is damaged, there is DNA repair mechanism in each cell that we have, right? But the damage, if it goes beyond, what will happen to the cell? The cell will go into apoptosis, right? Straight away cell goes into apoptosis, right? There is no future for that cell if the DNA is damaged, and everywhere that it is, right? Even in your body. Right, in your skin, right, if you have radiation injury, that means क्या होता है radiation injury, right? If you expose your hand to UV, what will happen, right? UV will damage your DNA so much that it will then the cells will has to go into apoptosis. If the damage is beyond the capacity that the cell can recover that damage, then it will, right? So that's what happened, right? Many of the people have exfoliation when they are exposed to the sun, right? Many people when they go sunbathing, what will happen to them, right? They would exfoliate, right? Unka skin just goes off, it just spills off, right? Sometimes you hear also some किसी को हो जाता है, right? वो क्या होता है? What happened to that case? That's basically a radiation injury, right? If radiation falls onto it, the DNA is damaged so much, right, that the cell has nothing but no fate but to commit an apoptosis, commit a suicide. Apoptosis is a programmed cell death, right? So it will commit suicide, right? It will not generate the subsequent generations with those kinds of damaged DNA, right? So DNA is so important for the cell, right? In nature also, right? Nature takes care of that okay, if the DNA has to be neatly protected, and when it is neatly protected, of course, when you have one covering, the second covering, double membrane, right, in the nucleus, and inside there is a DNA. Do you get anywhere into cytoplasm in eukaryotes? Is there any DNA into cytoplasm? No. No means except mitochondria has their own DNA, right? The mitochondria will have their own DNA, except mitochondria, right? There is no DNA. So you will, in eukaryotes, all the DNA is confined to the nucleus, right? You will find DNA nowhere into the cytoplasm. What will happen if you put a DNA into cytoplasm? Suppose I have a micro injection, I put DNA into the cytoplasm, what will happen? Right? There are only two fits. Right? What will happen to that? Exosomes. Right? I told you that enzymes, DNases, right? And then they will chew that DNA off. So they could be finished. Right? So there is no future for a DNA in cytoplasm. Right? Otherwise, the DNA has to enter into the nucleus. Right? If it enters into nucleus, then it will stand a chance that it will get fused with genomic DNA, and once it is fused with genomic DNA, then it is the genome, right? It's a part of its own genome, and then it is maintained, taken care of, replicated, everything happens good for it, right? That's what happens, right, in the genetic engineering experiments, right? What we try to do, we try to put DNA into the cell, right, and then we hope that the cell will uptake that DNA. Right, so uptake happens at two places. Uptake is through this cell wall, and uptake is through this nuclear envelope. Right, is right. So if it is like only into this cytoplasm, right, it will not have, does not have future. But unless and until it goes into nucleus, right, and then integrate with this own DNA here, then there would be a future for that. Right? So why we have to do so many experiments of transgenics? Right? If you have to get a transgenic, genetically engineered organism, what you have to do is you have to try and make many events, right? Create lots of events of transgenics, that successful transgenics, and then select one which is good. Right? Why we have to do that? Because not every time that a DNA you will put into will go into the nucleus. Right, and get integrated in its own genome. Once that happens, then only the transgenic experiment is complete. Right? Beyond that, what will happen? The DNA will get degraded, so nothing would happen. Right? Understand that? So that is where it will happen into the nucleus. So that's a basic difference between the DNA of prokaryote and eukaryote. Right? Another important feature is prokaryotes only have one single DNA, as I told you, right? But then in eukaryotes you have chromosomes, 1 chromosome, 2 chromosome, 3 chromosome. There are many chromosomes. Hundreds of chromosomes are there in certain organisms. What those hundreds of chromosomes are? As I told you, every chromosome is one single DNA molecule, right? That is most important. There is one most important difference between prokaryote and eukaryote. And what is that? This is haploid and this is diploid. What does haploid and diploid means? Haploid is one single set of genomic DNA, right? And diploid means we have got two sets of genomic DNA. So two sets usually eukaryotes have. Then they will have they are more stable, right, than having one, right? You always, when you do something important, letter or something on your laptop, what do? You always make a backup. है ना, कोई dissertation बना रहा है, तो what you will do? You will have a backup of that dissertation on a pen drive or a CD drive, like, or you will save it on some different place. Why you require that? Because if something happened to this file that you are using, right, you always can go back to the backup. When you have a car, you have extra tire. Why? Because if one punctures, right, you can always replace that with one which good one that you have, right, and then you can do away with that puncture at a later time. Exactly, right? The same thing happens with the DNA too, right? If I have one DNA molecule, one gene, say suppose I have one copy of one gene and that get mutated, what will happen? Then obviously I would suffer, right? But then if I have another copy of DNA which is good, then what will happen? I can always switch on to the another thing. So you can, like we spoke for a sickle cell anemia, right? If there is one mutation into that hemoglobin gene, right, then what will happen, right? That will lead to sickle cell anemia, right? But then I have two copies, so one better copy would produce better cells, and some cells will be sickle cell. So I would still have no much severe problem. But if I have both the copies mutated, then there is an issue, right? Then of course, then I will suffer with the sickle cell. Then I will be homozygous to that mutation, right? And that will be a suffering condition. Otherwise, I could be a carrier. But a carrier does not develop symptoms or develop milder symptoms. Depends on that, right? Depends on what disease we are talking about. But then it would be differentiable. So having two copies is useful than having one copy, isn't it? Right? So prokaryotes will have only one copy of their entire genome, whereas eukaryotes have double the copy. So better survival, isn't it? Right? So better survival tactics. There is one more important feature into that, and that is into their gene. Right? What is a gene? We define gene as the functional unit of the DNA. Right? That is what codes for something. That is what a gene is. So gene starts with the start codon, ends with the end codon. There could be some UTR regions, that is untranslated regions 3', untranslated region 5'. But then that is what a gene is. Prokaryotic gene, right? So that is what a gene is, like this. Like you have start codon, right? Here you have end codon. And then this is the 3', 5' UTR, and this is 3'. Right. UTR is what? Untranslated region. Okay. So this is what happens, and the start to end codon में क्या होता है? They will code for the amino acids, right? And they will make the polypeptide. This amino acid is actually a protein, which range from the start codon to the end codon. That is what a gene is in prokaryote, right? You have genes like that. They are undisrupted. But in eukaryote, what happens is it is interspersed with coding, non-coding regions. Right? Coding regions are what called as exons. Right, and in between us are what? Introns. Right? In eukaryotes, what happened? Coding and non-coding regions are. Right? I don't know if you have quoted this difference between eukaryote and prokaryote before, right? But in eukaryotes, the gene is interspersed, right? Or we call it as a spliced gene. The gene is spliced, right? It is interspersed between introns, right? And exons are together, which come, and then finally the a protein coding region is collected, and then that the protein is synthesized out of that, right? But in prokaryotes, nothing like that. It is like this. In fact, in prokaryotes, sometimes you have there are polycistronic genes. Means what? It's like a one gene here, right? Immediately another gene and a third gene. So one mRNA is produced, right? And then that one mRNA will make a protein, which then intersperse, and then 3 proteins are made. Right? We'll talk about that. Right down the line, we will see that in details. Right? So that is what happening in prokaryotes. There is one more difference in prokaryote and eukaryote. Right? In prokaryote, gene के पहले क्या होता है? What is this region? Before downstream to the gene is called as the promoter. Promoter region, right? And there are a lot of difference between the promoter region of this gene of prokaryotes and in eukaryote. Eukaryote also have promoter. They also have promoter. Right? And there are many proteins that go and bind to that promoter, right? And unless and until those proteins bind there, the gene will not express, right? So there are lots of such regions into this promoter of eukaryote and prokaryote alike, right? We will go into those details, right? We will in next coming lectures, but today you understand that there are differences into it, right? There is one region called as Tata box into it because it has TA repeats, so they are called as a Tata box, that is required for the RNA polymerase to bind to and then make the mRNA, right? So RNA polymerase will bind somewhere here and then transcribe it. So you will obviously have some UTR region, right? And then it goes into lamina. So you have a site where the RNA polymerase will bind and then transcribe the entire gene. Okay, right? So there are differences into it. One last most important difference, right? Because of these differences in their promoter, the prokaryotic gene expression and eukaryotic gene expression also differ, right? Eukaryotes have much more finer control on the gene expression than prokaryotes, right? What happens in prokaryotes and eukaryotes is it's like a very fine control and then a gross control. Say suppose like I put on this fan, right? So sometimes, and there is this regulator, fan regulator is there, isn't it? And it has like speed 1, 2, 3, 4, 5, usually 5 speeds are there, 5 is the top speed, right? And suppose if that is gone wrong, then what happens? This is the button besides this regulator also, that also controls the on and off status of this fan. But what happens is, right, suppose the regulator is gone bad, what will happen? The fan will be put on, it will go on to the highest speed, right? Or you have to put it off. There are only two possibilities, right? It is put on or put off, right? Either on a high speed or on the low speed. But then you won't be able to control the speed of the fan, and you know what happens with that, right? And then either you have to sleep with the heavy fan, take a rug and then sleep, right? Just because this fan just cannot, you cannot control its speed, right? So something, and then you have to keep adjusting with that, right? So either it is on at a higher speed or it is not spinning at all, right? So that's what will happen with the fan, right? And then with the regulator, what will happen? You can adjust it according to your comfort, right? The eukaryotic promoter is like that, right? It is like a fan with a regulator. So you can adjust the level of gene expression. Remember, that's very important point, right? Right? You will realize the importance down the line when we will learn many things down the line. But at least what you have to remember right now is that you have a good control on the system, right? It is not like on or off, or it's on the highest speed or off completely, right? So you have to have this kind of control. You require such control even on your car wiper, right? Car wiper may also have like 3-4 steps, right? And then accordingly how much rain is coming, you could either increase the speed or decrease the speed, right? And then you have this wiper continuously wiping your the front glass of your car. It also requires those kinds of control to adjust to your comfort, right? To adjust to their drive. That is what eukaryotes can do, right? What eukaryotes can do, they can control their genes expression very fine-tuned, right? Not gross, fine-tuned control. Not like on and off, but in between also. It's like black and white, or you have shades of gray in between. So if you have if you convert a picture into complete black and complete white, right, it doesn't look very good. But if you have shades of gray in between it, like half tone figures, right, what I am talking to, then the figure looks very nice. So you require that kind of control to make a very fine. And why you require that fine thing, right? You remember, no prokaryotes, right, can make multicellular, whereas eukaryotes can go into multicellular. That's the biggest difference, right? Biggest difference. One of the very important event in the evolution of life, right? That it turned from prokaryotes to eukaryotes. Right? Remember, we are all eukaryotes. All our cells are eukaryotic cells. We are multicellular organisms. Right? Right? Imagine if that would have happened in the evolution, the entire globe, right, would have been life, but all single cell life. Then the entire oceans would be a slimy place. Right? All bacteria, nothing can grow. Right? But because it grew into eukaryotes, it will grow into higher and higher forms of animals. Right? And that is only possible in eukaryotes. Why it is not possible in prokaryotes? Right? That's because they cannot control their gene expression. They cannot fine tune their gene expression. And if those you can fine tune your gene expression, two cells can come together, speak to each other, right, and can develop into multicellularity. Right? It's not simple event of multicellular. It's not like two cells coming and sticking up together each other. Right? That too happens in prokaryote also. They can form a biofilm. Right? You have heard of biofilm, right? That's what? It's an aggregation of prokaryotic cell. But does that mean it is multicellularity? Multicellularity means they further show differentiation in their structures, functions. Right? And we have talked about that. Your hepatocytes and bone cells and nerve cells and retinal cells will have the same genome, but they would behave completely different. Right? There is a good functional differentiation between them, and without those functional differentiation, you will not be able to have organs. Right? So you are basically, a cell would make into a tissue, multiple tissues will make into an organ. Right? Multiple organs make an organism, right? And multiple organism makes a population, right? Multiple population makes an ecosystem. Understand that, right? So that's like a hierarchy, right? And so you have to have a very fine control on gene expression, without which there cannot be multicellularity, and that is what is being seen, right? Multicellularity can only exist into eukaryotes, not in prokaryotes. Right? I'm sure you haven't quoted this difference between eukaryote and prokaryote before, right? But this is what is translates into it. Right? You had been just a laundry list of like 1, 2, 3, 4 eukaryotic feature, 1, 2, 3, 4 prokaryotic feature, right? But try to understand the meaning behind it. Right? So 3 important things that we talked about. Right. One thing is what? The between prokaryote, there is a difference in their genes. Right? There is a difference in their gene regulation. Right? And there is third thing is difference between their compartmentalization. And compartmentalization doesn't mean just a physical compartmentalization, functional compartmentalization. Understand that? Right? So that is most important. Right? So that is where we always try to look at. And the functional compartmentalization is important, right, to live an efficient life. Right? What's wrong between like living in one room apartment and living into 3 BHK apartment? Right? It's a comfort. It's not just comfort, but it's also an efficiency. Right? Tomorrow, one day you try to do it, take a stove into your bedroom and try to cook something. Right? Then you will see how inefficient it will be. Like you will put something, right? चाय बना रहे हो तो इधर चाय डालो, शुगर के लिए भाग के वापस किचन में जाना पड़ेगा. Right? The chai is boiling, now छलनी के लिए वापस किचन में जाना पड़ेगा, है ना? So everything would become inefficient. Right? But kitchen में करो तो क्या हो जाएगा? It will be so efficient. You know that which things are where kept. That map is perfect in your brain. है ना? So you know that where to reach for a matchstick, where to reach for a sugar, where to reach for a chai patti. है ना? So it becomes like more efficient. Right? In less and less time, you can make good tea. That's what efficiency is. Right? And that what efficiency you will see in eukaryotes. And that is absent in prokaryotes. Understand? And that efficiency translates into the multicellularity, which is only in eukaryotes and not in prokaryotes. Understood? Right? I think this is a very clear differences between the eukaryotes and prokaryotes, right? And you have understanding of that, right? Not just a list, right? Try to understand what does that, right? And when you understand that, you will be able to understand really more clearly, really more efficiently. Then you don't have to mug up things, right? If you understand that, okay, compartmentalization, then vacuoles भी आता है, then organelles भी है, then the nuclear and ये भी है कि Golgi operatives, all organelles, right, are membrane bound organelles. Yeah? Can there be organelles without membranes? Cannot be, right? So all organelles are there present in eukaryote, they are absent in prokaryote. That's because there are no compartmentalization, and compartmentalization is most important, right, for the efficiency of the cell. Clear, right? I hope I have made myself very clear, right? And